Hello everyone, welcome to my channel Love of Biology. Today we are learning about meiosis, nothing but the reduction division. The chromosome number, that is set of chromosomes, will reduces from diploid to the haploid, that is half of it reduces in the daughter cells. It is responsible for the formation of the haploid gametes, that is the male and female gametes. During the sexual reproduction they will form. So male and female fuse to give the zygote by fusion. Meiosis involves two sequential cycles of the nuclear and the cell division called the meiosis 1 and meiosis 2, but only one single cycle of the DNA replication takes place in the S phase. So in the meiosis 1, again it is divided into that is karyokinesis and cytokinesis. Karyokinesis involves the prophase 1, anaphase, metaphase and telophase 1. So in the prophase 1 of the meiosis, it is the longest one and more complex than the prophase of the mitosis. It has been further subdivided into following 5 phases based on the chromosomal behavior. Those are leptotin, zygotin, pachytin, diplotin and the diakinesis. In the leptotin stage, this is the first stage of the prophase 1. So where the chromatin thread will condenses to form the rod shaped chromosomes, and in the zygotin the synapses will take place. That means the homologous chromosomes will fuse together to form the synaptonymal complex. So in the pachytin stage the crossing over takes place, that is the recombination to produce the genetically variant offsprings. So the exchange of the genetic material takes place in the presence of recombinase enzyme. So next in the diplotin stage, the chase meta will form, that is the chromatid regions. They will separate homologous chromosomes and at the centromere they will bind to form the chase meta X-shaped structure. In the last step diakinesis, the nuclear membrane, all these will disappears. So in the metaphase 1, the bivalent chromosomes align on the equatorial plate. The microtubules from the opposite poles of the spindle attaches to the pair of the homologous chromosomes. In the anaphase, the homologous chromosomes will separate by splitting the centromere, while sister chromatids remain associated at their centromeres. Only the homologous chromosomes will separate. Whereas in the telophase, so here you can see the prophase 1 and in the metaphase, so the homologous chromosomes you can see, and in the anaphase and the separates. So after that telophase, so the nuclear membrane and nucleolus will reappear. Cytokinesis follows the telophase 1. Although in many cases the chromosomes do undergo some dispersion, they do not reach the extremely extended state of the interphase nucleus. The stage between the two meiotic phases is called interkinesis, and it is generally kinesis. It is actually resting phase between the meiosis 1 and the 2. It is followed by the prophase 2, a much simpler prophase than the prophase 1. So in between the meiosis 1 and the 2, the interkinesis is present. So here you can see the interkinesis. After that meiosis 2 will start. Again it involves the prophase 2, metaphase 2, anaphase 2 and the telophase 2. Let us discuss one by one. First prophase 2, which is a meiosis 2 resembles like a normal mitosis. So in the prophase 2, it is initiated immediately after the cytokinesis. The nuclear membrane disappears by the end of prophase 2. The chromosomes again become compact, and in metaphase 2, at this stage the chromosome align at the equator and the microtubules from opposite poles of the spindle get attached to the kinetochores of the sister chromatids. So later, coming to the next one, so here you can see the two daughter cells which are produced in the meiosis 1 will undergoing the meiosis 2. So in next anaphase, the splitting of the centromere of each chromosome move towards the opposite poles of the cell. The splitting will take place because of the spindle fiber will pull towards the poles. Next telophase 2, the two groups of the chromosomes once again get enclosed by the nuclear envelope. All the cell organelles will reappear. This follows the cytokinesis, resulting in the formation of 4